So we identified this gene as a candidate that we think is important in invasive growth, first by doing single-cell RNA sequencing of human brain metastases, where we sampled the middle of the metastasis as well as the surrounding brain outside of the contrast-enhancing area, where in theory, based on the current dogma, there really shouldn't be any cancer cells. And to our surprise, we actually found abundant cancer cells outside of the contrast-enhancing area in the surrounding brain. And when we compared these cancer cells that had invaded into the distant brain with those in the middle of the metastases, we found a large number of gene ontology and signaling pathways that were differentially expressed between these two groups of cancer cells, which together led us towards this one gene called cold-inducible RNA binding protein, which is a known protein with roles involved in the stress response that really works more mechanistically as an mRNA binding protein. And so it's a protein that's expressed in cells that essentially binds to mRNAs involved in overcoming stress responses and leads to their more efficient translation and expression that way. So we were able to show functionally with cohort of PDX models that we had, as well as our patient specimens, that this gene actually, in fact, is overexpressed in highly invasive brain metastases compared to the more minimally invasive brain metastases. And functionally, when we are able to knock down this gene in highly invasive brain metastases, we're able to decrease the growth of these brain metastases as well as diminish their ability to invade into the leptomeninges along the spinal cord in mice. So together, there is still quite a bit of work to be done, more mechanistically in discovering which specific mRNA targets are being bound and more functionally leading to this ability of cancer cells to adapt to the stress response in the adjacent brain. But we think that at least right now that it is functionally important in one way or another, whether it's actually leading to the actual invasion of cancer cells or is more important in the just allowing cancer cells to survive in the adjacent brain in a really inhospitable area surrounded by astrocytes and microglia and cells that certainly don't want them being there. Those are still some of the things that we have to figure out, but we're on the track that this gene is very important to this process one way or the other.